Welcome to this fourth in a series of Stanford Inventor webinars. Today we're pleased to have Dr. Tomek Shvaigut, one of the co-inventors, talking us today. And the topic of this invention is efficient stoichiometric delivery of multiple guide RNAs through cargo. Tomek is a senior research scientist in the Chemical and Systems Biology Department in the School of Medicine here at Stanford. He received his undergraduate degree at Warsaw University, his PhD from the Polish Academy of Sciences, and did his postdoctoral work in the Laboratory of Molecular Embryology at Rockefeller University, Cold Spring Harbor. His co-inventors are the principal investigator, Professor Joanna Wasaka from the Department of Chemical and Systems Biology and also of Developmental Biology, and also Bo Gu, who is a PhD candidate. So today, for the webinar, the first thing I would ask you in a moment is to just tell a little bit about your lab and what your lab does. But then we'll jump right into the slides to get more technical information about today's invention. And I want to talk for a moment about the question and answer process. So people can enter their questions at any time during the webinar. There are two functions that you'll find around the perimeter of your Zoom window. One is Q&A and one is chat. We're going to use the chat function. And if you want to submit a question, you do use the chat function. But please note that there are two ways to do that. You can either be sending the chat message to all panelists or to all panelists and all attendees. So if your preference is to remain anonymous, please use the all panelists option for that. And then toward the end, we'll open it up for questions and answers probably about 10 minutes before the end of the webinar. So with that, Tomek, let me turn it over to you.
Hi. So Wasaka Lab is interested in gene regulation and epigenetics in development. We are particularly interested in studying and understanding the function of enhancer elements. And to this end, I mean, we can be described as a basically basic biology lab. Developing technology is not our primary focus. So if I may have a slides, please. So in today's presentation, I'm gonna talk to you about a new, we think, exciting technology for improving CRISPR-Cas9 based methods. I'm gonna lead you through some motivation, how we arrived to this technique. I give you some technical details and also examples of real-world applications. This is either published stuff or stuff being in revision. And also I will discuss with you some additional potential for applications. So as I mentioned, Wasaka Lab is interested in how enhancers work. So enhancers are small DNA elements positioned all over the genome, quite often far away from the genes, but they contribute and regulate genes during development. They integrate information from cellular environment and from the outside environment. And our main focus is to understand how this activity propagates, what makes the enhancers, the mechanistic aspects, but also how enhancers and variation in enhancer sequence contributes to phenotypic variation in humans, between human population, between individuals, and also in disease. Can I advance to another slide?
Technical difficulties, just a moment, please.
Okay. So the technology has its root in our frustration with existing methods to interrogate enhancers in very chromatin context. So existing methods rely on fixed cell assays, such as ChIP-seq, meaning chromatin immunoprecipitation, which is a population assay, a conformation capture, which is also generally a population assay, and also with DNA FISH, which is single cell assay, but still relies on fixed cells. And we are wondering, can we visualize enhancers in live cells? And this would allow us to answer a lot of open questions in this field, such as what is the dynamic movement of enhancers and promoters in living cells? Is it dependent on the activity? Can we infer activity from this motion? And also, what are the relationship between the enhancers in the 3D space? In the past, work by many labs established that exogenous and endogenous repeat sequences can be visualized in living cells. And the oldest technology of this kind, and the most robust one, relies on integration of large arrays of prokaryotic operon repeats, such as LAC-O or TET-O repeats, which can be then visualized by recruitment of the operon proteins, such as LAG-I or TET-R fused to fluorescent proteins. So this works very well on the microscopy level, however, has at least two really serious drawbacks. One is it's very cumbersome to apply it to different loci because it relies on homologous recombination. It takes forever to develop the reagents. Secondly, the arrays are very large. They dwarf the elements you want to visualize. And because of the repetitive nature, can induce epigenetic changes in the chromatin. So with the advent of CRISPR-Cas9 methods, lab of Stanley Chi and Bo Huang developed this method of visualization based on CRISPR-Cas9 approach. So if they fused the Cas9 to GFP, and if you recruit it to the repeats, like in this example, to the telomeric repeats, you actually get very beautiful labeling. But telomeres are very easy, or generally repetitive sequences are very easy to image because with a single guide, you can actually deliver up to 2,000 molecules to a single instance in the genome. So can non-repetitive sequences be visualized? And it turns out it's not that simple because in order for typical microscopy modalities to actually distinguish the loci, you need about estimated 25 copies of fluorophore present at the locus. And Stanley Chi group tried in the paper and demonstrated that in principle it is possible to image non-repetitive loci by delivering a large number of gRNAs. In their example, it was 73 gRNAs. However, I mean, it was published in 2013 and it never caught on. And even when you look in the paper, you see that the labeling is very inefficient and signal to noise is pretty bad. So we were wondering, why is that? Because in principle, this should work actually really great. And we think the major reason behind it is need for uniform delivery of many gRNAs to a single cell. And what I mean by that is illustrated in this slide, which is a result of a mathematical modeling of the delivery of multiple molecules to the cell. So here we have a situation where we try to deliver 36 gRNAs. On x-axis, we have multiplicity of infection, and on y-axis, we have how many of given molecule actually a single cell got. And as you can see, efficient delivery of that many molecules requires some biologically unrealistic multiplicities of infection. So even with 100, multiplicity of infection 100, with most delivery methods would actually kill the cells, you still get gRNAs which are not delivered. So the blue dots on this, I highlighted in blue the stuff which doesn't get transfected. So for comparison, the same data are drawn to scale with a situation when we would have a construct which can deliver all the guides on one, basically present in one construct. So you see that it completely dwarfs the situation of co-transfection or co-infection, just underscoring how inefficient, in fact, delivery through co-transfection or co-infection is. So we surmised if we could actually construct arrays of expression units which produce gRNAs, we could overcome that serious bottleneck. So it is well established that RNA pol-free transcriptional units are excellent method for expressing small RNAs. So such unit in this case consists of the promoter, the gRNA itself, the gRNA scaffold, and a terminator sequence. So when you think about repetitive array of such elements, you will notice that the two adjacent units, gRNAs, provide convenient unique breakpoints from which unique recombination arms or unique sticky ends can be derived, which can form building blocks from which to build such an array. So such a building block would have halves of gRNAs, and in between a constant region, which basically is a permuted expression unit. So how we get to these building blocks? And this is basically where the method really starts. So we start with a constant fragment, which is this permuted unit, and this is a recombinant DNA fragment, and hybrid synthetic oligos, which consist of the two halves of adjacent gRNAs interspersed or disrupted with the type 2S restriction sites. So these two molecules are ligated to form a mini circle, and this mini circle is resolved with restriction digest to produce the desired building blocks. And then the building blocks can be ligated together with the vector to form the final array. And we routinely construct arrays of n's 12 to 18. Higher are possible, it just the efficiency goes down a little bit. These arrays allow for uniform stoichiometry of delivery. These are parallel expression units, which actually make the expression very efficient. They don't rely on additional processing, so you don't have bottlenecking from saturating some processing pathways. In fact, they actually are a natural occurrence in human genome, so they are not that unusual, at least in eukaryotes. The actual construction process is rapid. It takes about 2 to 3 days. And interestingly, our biggest worry originally was this stuff is not going to be stable. And in fact, they are stable both in prokaryotes, so you can very easily propagate them in typical bacterial strains, and also stable in vivo in mammalian cells. So I'm going to walk you through the first application of this method. So we were actually interested, can we actually visualize the loci in vivo? To this end, we constructed a mouse ES line expressing DCAS9 GFP, and we transfected a 36-mer cargo array targeting vicinity of the enhancers of FGF5 gene. So as you can see here, upon transfection of the cargo array, we see this dramatic relocalization of DCAS9 GFP from nucleolus to nucleoplasm, an appearance of two bright, very contrasty spots, which correspond to the FGF locus, and we have proved it both through chromatin IP and DNA fish. So you see a perfect colocalization with DNA fish in this slide. And to our surprise, the approach was so robust, we can actually live track the loci. So as you see here, we have two cell nuclei labeled with cargo GFP for FGF5. This cell is in a so-called ESC state. In this state, the gene is not expressed. This is APLC state in which the gene is expressed. And if you look carefully, you will see that there is a much more motion in the APLC state correlating with the gene expression. This approach is robust enough that for our first publication, we were able to track about 2,000 cells, which was, I think, the first time anybody did it in eukaryotic cells at this volume. We also constructed many other cargos for different loci. This is an example of a cargo for TBX3 promoter. On the left, you see ESC state. TBX is actually expressed in this state. And again, you can see that the motion of the loci is much more rapid than in the APLC state where TBX is not active. Cargo approach to imaging is so robust that you can also use it for live fixed correlative studies. So in this example, we tracked first loci live, recorded their trajectories, then fixed the cells and come to the same cells with a fixed cell approach, in this case, single molecule RNA fish, which allowed us to correlate the presence of a nascent transcript with increased motion of the locus. And here I'm going to briefly touch upon some unpublished stuff, so I will not go to great details on it, but I will also demonstrate to you that Cargo dCAS9 improves CRISPR interference-based method to a great extent. So CRISPR interference was also invented, I think, in Stanley Chi Labs. The approach is to fuse transcriptional effector domains such as VP16, which is an activatory domain, or CRAB domain, which is a repressory domain, to a dCAS9, meaning catalytically inactive Cas9. And upon delivery of these molecules to a target such as promoter, we can exert either increased or decreased transcription from this target loci. And it was kind of known in the literature that single gRNAs don't work that well. It works, but it's not a really high modulation. So what we did here was to design a 12-mer cargo array targeting LTR of a retrovirus. And we're talking here about divergent retrovirus. So actually, we're not trying to modulate a single lab strain. This is actually a wild sort of population of virus with different differences. So the idea was to design a minimal array which can conceivably target almost everybody in the population. And on the left panel, we see results of the expression analysis of the viral genes for a CRISPR-A, which is the activatory CRISPR interference. And as you can see, the targeting cargo array, but not the non-targeting cargo arrays, increase the expression of the viral genes about 6-fold. And you have to realize that LTRs are already very active elements. So we just boost it further 6-fold over the basal high activity level. Even more excitingly, we can achieve a complete repression of the viral genes. So we actually can bring down the expression of the viral genes to the undetectable level using this approach. So in summary, what I presented you today is that Cargo allows for rapid synthesis of arrays expressing tens of gRNAs and allows for efficient delivery of stoichiometric mixtures of gRNA, and such optimized delivery allows for efficient visualization of non-repetitive chromatin loci in live cells, and also that Cargo boosts the efficiency of CRISPR-I or CRISPR-A-based interference technologies. Finally, I want to touch upon future applications. So this is a very general approach. You can use Cargo also to assemble siRNA, shRNA, microRNA arrays, and also construct mixed approach where you have a siRNA mixed with dCAS9 approaches. It can also be used to boost expression of a single species of RNA. So Cargo can be constructed from identical gRNA units, up to about 25 to 36 elements, which basically will bring the expression of this RNA, if desired, 10-fold to 30-fold. It also has a potential to increase the efficiency of genome editing, particularly for producing knockouts, because you can deliver multiple interfering RNAs for the same gene at the same time. As I shown, it increases efficiency, but also it can increase the specificity of CRISPR-I and CRISPR-A methods by diluting the off-target effects. It is useful for broadening the spectrum of targets, which I think is a very interesting potential future approach. Basically, you can try to checkmate the evolutionary pathway of the pathogens so they cannot escape the interference. And thank you for your attention.
So we'll jump to the Q&A in just a moment, but we're going to dwell on this slide for a second so that you can write down any of this information you're interested in. And in particular, if you're interested in learning more about the technology or the possibility of licensing it, please contact Chu Chang, and there is Chu's email below. She's the licensing associate responsible for this technology. So again, we're going to use the chat function and invite people out in the audience to submit any questions they have. By sending it to all panelists, you'll avoid having anybody else see your identity if that's your choice. So until we get some from our audience, let me just ask you, Tomek, can you say a little bit about the cost? If one were to do this and do it regularly, what sort of cost would one be looking at?
So in the lab environment, basically using retail prices, the costs are between $200 to $400 per array, depending on how large it is. And the major, about 70% to 80% of the cost is actually DNA synthesis. So basically, if you do your own DNA synthesis, can purchase reagents in the bulk, this can be easily shot. I mean, you can at least decrease the price by at least 50%, I surmise, not more.
Okay, great. Let's see. So again, I'd invite the audience out in the internet land to submit any questions you have using the chat function. Don't see any quite yet. Let me ask you another one. What do you see as the leading applications in terms of, you know, volume of work, volume of tests? If a company were interested in licensing this and then, you know, selling the test capability, what do you think is the biggest application?
I think right now the biggest application would be to actually CRISPR interference. I think it has a potential to take over the market, which used to be the siRNA market, short hairpin market. The reagents are orders of magnitude more powerful in terms of the gene manipulation. And also, if anybody looks for possibly therapeutic approaches, the CRISPR interference methods are safer than actually having an active nuclease present around. And with cargo combined with CRISPR interference, you can achieve the reduction of expression comparable with the knockouts.
Another question might be, you know, I know you're not a patent attorney, but just to share your thoughts about, you know, this technology and whether one could use it without licensing a bunch of other intellectual property from others, or I mean, is it really distinct, or does it build on existing patents? What's your perspective on that?
So, as I say, I'm not a patent lawyer, and I actually have very little knowledge about this. But the core of the technology is basically an assembly procedure. So I think, especially if you provide it as a service, like basically we construct for you the arrays, I don't think this runs into that many additional intellectual property conflicts.
Okay, great, thank you. So one last call for people from the audience. If you have any questions, please use the chat function. I'm not seeing anything yet. So we'll give that another second. And maybe what we'll do now is we'll switch back to the last slide so that people can jot down the information if they didn't have a chance to do so already. And again, there's the docket number for the Stanford invention. There's a link to our TechFinder website where you can actually also search for many other inventions by keyword. And again, the licensing associate responsible for this is Chu Chang. So with that, if there are no more questions, well, I guess we'll close the webinar. And thank you, Tomek, very much for speaking to us today. And thanks, everybody, for attending.